Yes, good evening to everyone. So, welcome to the last day session, that is on 14th day session on AI and ML approaches of drug and vaccine design. So, this is our theme, and till now, whatever we have discussed from day 1 to day 13 is is related to drug and vaccine design. 1 minute, I can close the WhatsApp so it troubles us. Yes, let me continue the session. So today we'll be discussing on the major part of artificial intelligence and machine learning approaches of drug and vaccine design. So just a slide on my kind of a supervisorship. So I'm an approved Anna University PhD supervisor, and this is my reference number. And these are my areas of research: drug design, vaccine design, pharmacognosy, and immunotechnology. So three students have registered PhD under my guardianship, and even I am co-guiding students in Madras University. So these are the today contents. So today we'll be discussing on what is machine learning and how what are the applications of machine learning in drug discovery, and we discuss on a software MATLAB in drug discovery, and then we discuss how about AI, artificial intelligence, and how about its role in drug discovery. Then we discuss on tools and databases in drug discovery. We also be discussing on Python coding for machine learning approach in toxicity prediction. Like these two case studies are done by two students. One is from a UG student and another is from a PG student. Okay, let me take you to the machine learning approach. So, machine learning is a field of study in artificial intelligence. So, artificial intelligence is a overall field. There we can see this machine learning, and if you see, this is concerned with the development and study of statistical algorithms that can learn from data and generalize to unseen data. So generally, we use machine learning for inactive compounds. Generally, if you see the drug design, we see the ligand-based drug design, structure-based drug design, and if you see this ligand or structure-based design, we need the structure of a ligand in ligand and structure of ligand and receptor in receptor-based drug design. So where if you have an approved drugs, okay, whatever drugs used in market for treatment of disease, or what we call it as an actives, and we also classify the compounds as inactives. These compounds are not existing in market for treatment, but these are newly developed compounds. So if we want to work on a screening of compounds with these kind of inactives, then even we can bring such a kind of a machine learning techniques. There are multiple applications of machine learning. We see one by one. First, we see about the machine learning. Arthur Lee Samuel, an American pioneer, is the father of machine learning. So you can see this picture, and this is him, and he is an expert in the field of computer gaming and artificial intelligence. If you see this machine learning algorithm, we can classify the algorithms as supervised learning, unsupervised learning, semi-supervised, reinforcement learning. And the supervised learning, we can see the learning tasks like classification and regression. So these are some of the methods of supervised learning, and these are some of the methods of unsupervised learning. And similarly, for reinforcement learning, we can see this kind of a machine learning techniques. Now, if you see this machine learning in drug discovery, where exactly we can fit the machine learning in drug discovery, we can find materials, consumables, like what exactly we can use for drug design and chemical synthesis, combinatorial synthesis in medicinal chemistry. So there we can use the machine learning, and we can see the data mining, and wherever we want to develop the ontology, or even if you are seeking ontology information, and similarly biology research where we focus on target validation, and then we can discuss even in lead discovery and preclinical development, and we can also see in biomarker discovery, clinical development, pharmacovigilance, drug repurposing, virtual lab assistance. So these are some of the applications of machine learning in drug discovery. So if you see the machine learning in supervised learning, okay, we can see it basically two different methods. One is classification, the other is the regression. In classification, we find these four methods and the applications of these methods we can see here. Okay, like if you see the classification, this can be used in target disease drug associations and target drug ability based on pharmacokinetic properties and protein structure, tissue specific biomarkers from gene expression signatures. So the regression analysis we can see it can be used for these machine learning techniques can be used, and these are some of the applications. So more about this we can see plenty of journal papers on the applications of machine learning in drug discovery. So now we can discuss on machine learning system arrangement. So if you see this machine learning, initially we have training data set data. So the training data set data we develop from numerous data. Like if you want to check an activity of a compound, okay, like generally the activity of a compound is determined through QSR analysis, isn't it? The QSR is a ligand based approach. So if you want to go with checking an activity from nearly 50 compounds, okay, we can develop a kind of a training data set with the 50 compounds, and whatever activity, this for 50 compounds, the activity should be known. If you know the activity, we can post it in the training data set data. And if the activity of unknown compound has to be predicted, like a 51st compound where the activity is unknown, that can be predicted from this training data set. So whatever the one which I'm going to check the activity is what I call it as a test data. Okay, so I can check the test data from the training data set. And if you see this kind of a machine learning algorithm, we generally not only use restrict up to the ligand based, like building a training data set and using a test for checking the values. We can also use for multiple applications that even in a structure based drug design, we can use this machine learning arrangement. And if you see this, so participants can join on time. I don't know, some problem is with the Google Calendar. The Meet link was deleted when I tried giving an extra time on yesterday. Okay, so join on time. Okay, let me come across commonly used machine learning software and tool. So the tools what we can use is Theano, Keras. The Theano is actually a Python framework for fast computation. Keras is using the Python deep learning library. The third Torch, fourth scikit-learn, fifth Python brain, sixth TensorFlow, and seventh R, eight Vika, and nine MATLAB. Okay, so these are some of the machine learning software tools we are using. And if you see these kind of initially the one to this, okay, is generally using this Python programming code, but the R is something different where we can wherever we need statistical computing, we go with R. And this is of course a data mining tool in Java, developed in Java. My mentor, Professor Michael Kromiha, who is a pioneer in protein bioinformatics lab in IIT Madras. So he is teaching these kind of courses, 2 days courses often, so once in 2 months. So try to expertise by enrolling this kind of a course. This is also available in NPTEL SWAYAM. And about the MATLAB, it is a kind of a commercial software. Fortunately, the SVC has a MATLAB to work with it. So MATLAB has its own unique language, MATLAB programming language, where we can use this machine learning approach. Commonly used machine learning software and tool. The drug design applications in AI. Okay, if you see that in drug discovery process, for target identification, where we see prediction of PPIs and prediction of protein folding. Okay, so wherever we find these kind of a drug design topics, so there we can use the artificial intelligence models. I told you, machine learning is a part of AI. Okay, so more about AI we will be discussing in subsequent slides. So this is a slide mentioning about the applications of AI in overall. Okay, so not only you can see the AI involvement in target identification, we can also see this application hit discovery in terms of drug repurposing, like whatever already existing drugs for treatment of a virus if you find a new virus strain is coming, we can use the old drugs for an new upcoming viruses. So this is what drug repurposing I have mentioned earlier. Virtual screening, okay, screening of multiple ligands, and activity scoring. For all these things, we can use AI models. So if you see, especially virtual screening, if you download a data set of 50 compounds, and if you try to dock with a target receptor, so whatever you can able to dock with one ligand with one receptor, the same thing will be followed from second to 50th ligand. So how is it happening? Still the AI is developed inside. Okay, AI is enabled there. So in such a way that after the execution of each and every ligand talking with the receptor, it proceeds with the immediate ligand. Okay, like this, we can able to complete all the ligands whatever we use in the Autodoc Python RX program. So here there are, so you can see PYRX is nothing but Python RX programming. Okay, similarly we can see the AI is already developed in the existing softwares, in few softwares, and if you want to use the machine learning to develop our own models for newly synthesized compounds, we can still develop through the coding like Python or programming, MATLAB programming, etc. And also you can see a application a hit to lead in case at de novo drug design and even in lead optimization. Okay, so how the admit properties we get from different databases. Also, it works on these neural networks, multitask type of neural networks. Now, related to the machine learning classifiers, okay, so each and every type of cancer can also be predicted on machine learning classifiers. The only thing is that we take these samples, okay, whatever samples we get positive or negative. You know, the positive and negative samples are available in various public domain databases in the form of data sets. For example, in John Hopkins University, out of 168 samples, you can see some 90 or infected and the remaining or uninfected with breast cancer after diagnosing. Okay, so like in John Hopkins for a particular 1 or 2 months, how many patients are coming, how many are diagnosed, how many are with the positive, and how many are diagnosed with the negative. That is I won't get. So all these data sets, okay, not only from particular John Hopkins, they can go with the Mayo Clinic, go with the Anderson Clinic. So whatever data sets we have in various numbers, we can put in kind of a support vector machines, okay, and then we try to classify the data. Okay, for example, if positive breast cancer is shown in green color and the normal patient, the patients not diagnosed with breast cancer shown in red color, we put it in the form of a SVM and try to classify the kind of a data. And similarly, we can go with the nearest neighbor to check even the expression of breast cancer patients. Okay, like what is the expression of the breast cancer, and what I mean, what is the stage of a breast cancer for the disease patients. So all these things also we can further subclassify the data. Okay, so like whatever we can see the classification, regression, all these things we can try to use even for breast cancer prediction, like prognosis, diagnosis, each and everything can also be distinguished using the various algorithms. And whatever computational methods in drug design we have discussed, okay, if you see for space, not only for SARS-CoV-2, even in general, whatever we have discussed, all these methods, of course, these methods we find machine learning techniques are enabled there so that we are utilizing these kind of tools. And if you see this drug repurposing, in drug repurposing, also we see this application of machine learning majorly. And if you see in ANML approaches, we can see for prediction of repurposed drugs for coronaviruses. So, like for coronaviruses, for if you want to predict drugs, okay. So even how the data set is deposited in NCBI, if you try to download a data set from NCBI, we can get around 2,000 compounds, and these compounds are deposited only through the kind of the machine learning algorithms used in that NCBI server itself. So like they've used support vector machine, random forest, artificial neural network, nearest neighbor, deep neural networks. So these kind of machine learning algorithms are used for model development, and even the data sets can be created. Okay, not only for drugs, but also for inactives, whatever repurposing molecules we can able to use. Okay, so generally we can use the type all the types of machine learning techniques, of course, for prediction of repurposed drug for any type of diseases. Now we can discuss each and every machine learning technique and an example supporting the machine learning. First, we discuss on the support vector machine. If you see in the support vector machine, okay, if I want to classify drugs as lipophilic, okay, and hydrophilic, so lipophilic and hydrophilic, okay. So now check this kind of a tool. The example I provided is Swiss ADME. So in Swiss ADME, this is actually a boiled egg diagram. If you remember this, this is a boiled egg diagram. In this boiled egg diagram, if I draw a structure, for example, if a drug is a plant compound, okay, I just fed a plant compound, my curcumin. So whatever curcumin I typed, okay, it's lying somewhere in this white region. According to the boiled egg diagram, so the white part you can find majorly albumin, that is a protein. Major of proteins you can find here, but major of lipids can be found here, isn't it? It's an yolk sac, and generally you find high lipid content. So if our drug is locating somewhere here, this is a lipophilic kind of a drug. And if our drug is lying somewhere in this white region, not coming out of the yellow part, then it is an hydrophilic drug. Okay, so the curcumin is kind of a hydrophilic kind of a drug. So only we can able to find the location of our particular drug. Similarly, if you have a numerous drugs in the data set, for example, nearly 1,000 compounds, and if I want to check what are hydrophobic and what are hydrophilic, I want to distinguish the two compounds. Okay, we can use the support vector machine. So we can use a kind of a plane, and we try to distinguish the hydrophobic. Might be the hydrophobic is in red color and the hydrophilic might be in the blue color. We can distinguish these two different colors based on this support vector machine. Actually, the support vector machine uses a data classification algorithm which is used for data normalization problems. The so-called hyperplane, this is what the blue line, whatever you see, a kind of a random line. Okay, this is a line that separates the data input nodes with different values, and the vector from this point to the hyperplane can either support it. I just told you and with an example, isn't it? So just you can get the same meaning from there. So, but one thing you have to remember, we get a kind of a best hyperplane would be the one with the largest positive vectors and separating the most of the data nodes. Now let me take you to the next machine learning technique. The next machine learning technique is the deep learning. So in deep learning, deep neural networks. Okay, and for this we can see generally how what is first of all neuron and what is neural network. We should understand first about that. Then we can understand about the deep neural networks. Okay, actually, what is a neuron? Actually, according to the biology, this is actually a functional part of a brain, isn't it? This is a functional part of a brain. The brain is made up of billions of neurons, and whatever neurons we have, okay. So if this is a dendrite structure of a neuron, and if you see a neuron in computational structure, neuron in a computational structure, generally we have something like a CPU unit where we can feed multiple inputs and we can get an output. So using the multiple inputs to calculate an output, we use a kind of a correlation. And if you see in the form of a neural networks, so just like not feeding an input and getting an output from a cell, but the cell whatever we construct, that's what we call it as a neuron. Actually, the biological neuron distinguishes from the computational neuron, okay, in receiving the inputs and what output we can get. Generally, what the neuron can do in biological. So generally, if the acetylcholine can bind with the neuron and it can give the stimulus, isn't it? The stimulus is what we receive. Like whatever, if you touch a hot body, we immediately receive a kind of a stimulus, isn't it? So whatever input we receive as a heat, okay, high temperature, okay, so we try to recognize that we are getting a high temperature, isn't it? So that's what we can able to make it using the computational units. In the computational unit, in between the input and output layer, we can also have numerous hidden layers. And then what is about the deep learning here? How the deep learning comes into this kind of multi-layer neural network? The deep learning neural network are among the most widely used AI and machine learning algorithms. This has been used widely for analyzing medical images. If you want to check the pattern of MRI or pattern of the X-ray, or then if you want to come up with a kind of a conclusion whether the patient is diagnosed with a particular disease or not, okay, the computer can able to analyze through various colors and various patterns and intensity of colors. So all these things, okay, the intensity of color can be a hidden layer 1, and the colors, the type of colors can be a hidden layer 2. Okay, and the patterns, what kind of a patterns you observe, so that can be a hidden layer 3. Like this, we take multiple hidden layers to identify, to diagnose the kind of a disease after taking from medical instruments. Okay, so this is where you can find the deep learning models. And if you see the third machine learning technique, this is the k nearest neighbors. Okay, k nearest neighbors, the best example we can consider is the Jing 15 docking tranche. Okay, so we have Jing 15, Jing 20. So there are different type of docking tranches. You know what do you mean by tranche? The tranche is something like if you have billions of compounds, whatever billions of compounds, or we have major of inactives and also we have actives. Okay, if you want to get actives alone, we can get an active data set. If you want to get both, we also can get both. So whatever I mentioned, there are billions of compounds in a single database called Zinc. Okay, and if you want to classify these compounds in various categories, okay, like keeping a molecular weight in Daltons from 200 to 500 and above 500, and even partition coefficient from -1 to 5 and above 5. Okay, so in this, I told you even during the virtual screening, if I want to download the data set of compounds where the molecular weight is 350 and partition coefficient is 3, for example, I'm mentioning. If you want to download the molecular weight of 350 and log partition coefficient, so this is the data set. We can find around 370 lakh structures are available. So if you download these structures, okay, you can use it further for visual screening and or other purposes after the visual screen. Okay, so in k nearest neighbors, one can able to find what are the nearest neighbors for the classification. So if I, so here I just round off this. So whatever I round off, the nearest data, nearest drugs, whatever we can get, we can able to download it. Okay. So here you can find the application of this kind of a machine learning technique. The fourth is the random decision forest. Okay, in random decision forest, we can say an example like Galaxy QSAR. The random decision forest are formed of decision trees where multiple samples of data are processed by decision trees and the results are segregated. Okay, so generally in QSAR, especially I've given an example like an online QSAR called Galaxy QSAR. We use this random decision trees for classifying the data in the training data set, and so the test data set value can be we can able to get the value for the test data set. The next is the fifth one, I believe. It's a neural network. I already come across the deep learning. Now I just mentioning about just the neural network alone. So if you see this neural network, okay, so if uh, uh, if we want to diagnose the COVID-19 patients, okay, we have to first initially build a kind of a training data set through machine learning technique. So whatever training data set we are going to develop, okay, if we get an input from the X-ray images, generally how the COVID-19 can be diagnosed. So we have to take X-ray in the lung, isn't it? So in the lung, so here you see the much of the lung is invisible. You can see much of white portions here in this image when compared to the previous. Here you find less white, but you don't find any white images here. So this patient is not diagnosed with COVID-19, I can tell, but this patient has some kind of an infection. Okay, and this patient is having much infection. Might be he is infected up to the eighth day, where the SARS-CoV-2 infection is aggressive. So it is blocking the kind of a lungs information. Okay, so like this, if you get a kind of a data, how we can able to find it through that white, black, and gray regions. Even through gray regions, we can identify whether the COVID-19 infection is present or not. Okay, this we can able to determine through neural network, and not only just using this white, gray, and black images, but we can also, whatever images we have, we can create in the form of heat maps. In heat maps, wherever you find more infection, you can find in this form of red and yellow. If you form this kind of a red and yellow, this is more infectious. This data is more infectious. Like this, so through the colors, we can able to identify the infection. Okay, just like that, we have to send it through the softwares where the heat maps can accurately predict when compared to this. Okay, so these kind of techniques we can able to make through the neural network, and generally the neural network can be practiced through the NIME, TensorFlow, VCAR, Rapid Miner. Myself, I practiced with the Rapid Miner. Okay, so it's of course a commercial software. Okay, but it is plenty of work we have to make. So many PhD scholars can take this as a PhD research for diagnosing, for creating a kind of a machine learning algorithm and diagnosing the samples from the hospitals. So this is like a kind of a PhD research, and I'm showing an example like how we can able to make with R programming and machine learning. In our programming, if you want to screen the drugs, if you want to select a particular drug, okay, we have to load the libraries. We have libraries like RCDK, chemometrics. Okay, so each and every library you can find so many functions. We can utilize so many the functions, like if you want to load the molecules, okay, we can use the chemometrics, and if you want to view the molecule, we can use another library. Like these are some of the packages. The packages have numerous functions to visualize, to screen, to determine the activity, to show fingerprints of the particular molecules, to show the graphs from whatever compounds we have. Okay, so we have to load the libraries, and then we have to use the functions, and using the functions we can able to show, like even if you want to determine ADME characteristics, we can determine ADME characteristics after loading all these molecules in data set, and we can go with a few more functions, and final function we use it for listing the ADME properties of a particular compound whatever we have selected. Okay, so in R programming, we have plenty of packages, and we have find the functions inside to utilize the drug design concepts. And now let me take you to the next MATLAB. So MATLAB is a commercial software. Actually, even in MATLAB, we can able to see all the supervised and unsupervised learning. Whatever we can see the general concept in machine learning and general methods, we can find everything in this MATLAB. So all these methods we can see: naive Bayes, nearest neighbor, decision trees, linear regression, hidden Markov model, neural networks. So about the MATLAB is also linked with the TensorFlow. The TensorFlow is something linked with cloud computing. If you have large number of molecules and if you're working with the projects, okay, obviously we need these kind of a TensorFlow, okay, where you can link with this MATLAB. Both TensorFlow and MATLAB are widely used for the deep learning. Many MATLAB customers are interested in integrating TensorFlow models. And more about this, we can also see from the blog. Anyhow, I'll be pasting these blogs in the WhatsApp web. You can watch through that. And if you want to learn about the MATLAB, we have plenty of online courses posted in this MathWorks. The MathWorks is what they're providing this kind of a software called MATLAB. Okay, for AI, machine learning, and deep learning, we have nearly 6 courses. Okay, from basics to the advanced one can able to work on this AI and machine learning approaches. This is applicable for all the disciplines. Anyone can learn and anyone can practice with it. Okay. Fortunately, we have in SVC, each and every student has an individual license for the MATLAB, including the faculty members in our institution. And now machine learning using MATLAB online courses. Even we have something like online courses to secure the certificates, like how you find Coursera, Udemy. Okay, so like this we can find the courses, online guiding courses, and even you can secure the certificates after the completion. If you have the license version, I think this is a free one can try, and you can also get the kind of a certificate. Okay, there are plenty of courses available. The best can start with machine learning with MATLAB. Now, if you want to learn in freeware in online tools, I suggest you to go with the W3 schools. So in W3 schools, we have machine learning. So one can able to learn the machine learning from Python using this website called w3schools.com. Now let me come across artificial intelligence. Just now, whatever I covered is something about machine learning methods, the application of machine learning in drug design, and then I've come across different methods and its application in each and every database and tool developments. Now, let me come across with artificial intelligence. Artificial intelligence, I told it's a broad subject, and machine learning is one such subject from AI. Okay, I already showed you in the previous slide. So if you see this AI and ML is of course an integrated approach, and you can tell it is a software program that has been trained on a set of data to perform specific tasks like recognizing certain patterns. For example, if you have a series of lung images for the COVID-19 patients, and if you want to train a data set, and from the lung images, one can able to check whether he is infected in day one or he is infected up to day three or day seven, day eight, whether he is in the quarantine period or he is going to end the quarantine period. So everything we can able to determine only from the images. Okay, can be taken from the X-ray like that. Okay, so whatever we train a list of data sets, and if you find a new patient is coming to the hospital, and immediately if you get a kind of an X-ray, if you just try to find what day infection, immediately can able to find from the training data set. Okay, so not only we can take a kind of a digital data, we can also take analog data for analyzing the patterns. And if you see this list of the most popular AI models, so whatever we see this machine learning techniques, okay, of course we can use it for developing AI models. So father of AI is varying from father of machine learning. So father of AI is John McCarthy. He's an American computer scientist pioneer, and he play a major role in defining the field development to the development of intelligent machines. Okay, so this is about his alma mater, and related to AI. I told you AI is a broad subject where machine learning is one of the subject of AI. So we can use the types of machine learning techniques and sub techniques to drug design and discovery. I think I already showed you in this slide that the drug design, okay, for all this drug discovery process and all these drug discovery topics, we can use the types of AI models. Whatever is popular kind of AI models used in development of softwares that are all shown here. Okay, so the expansion of this abbreviated words are shown here. So you can find from the publications. I can also send you the publication link to get more information on this. So applications of AI in drug discovery and development. If you see this applications of AI, it can be used for pharmaceutical product management. Okay, in these applications and can be used for drug discovery, can be design and screening, and pharmaceutical product development. Okay, so aid in deciding suitable excipients, monitoring and modifying development process, ensuring in process, and similarly pharmaceutical manufacturing can be used in quality assurance and quality control, can also be used in clinical trial. So almost the AI is used in multiple applications, okay, in drug discovery and development. So artificial intelligence for drug discovery. So we can also use it for biomarker development. If you want to diagnose a disease, what is the most important protein target, and what are the best drugs for the protein target, and what are the interacting proteins for the protein target. So like this, so many studies, okay, based on a particular biomarker can be studied using this AI. Okay, for this we can integrate the omics data, chemistry data of the molecules, biology, that is biological data of the target protein receptors, imaging data based on the disease images from X-ray, MRI, clinical data, like whatever doctor take after giving a drugs, whether the patient is curing or not after third day, after fifth day, after seventh day. So the doctor's data, okay. So that is what clinical data for the treatment of disease, metadata. So all these things can be integrated, and we can use this machine learning techniques, and we can come up with a kind of a solution. I just in an interface, we just ask all this information, and we can from that we can get a final drug what can be used. Nowadays, in BPO, we can able to get the drug, okay, just by interaction, isn't it? Or by just going to this AI-enabled software. So this happened only through the AI. The involvement of indulgement of AI brings a kind of screening of or determination of a drug for a particular disease with multiple illness. Okay, and now if you see with this in silico approaches in drug repurposing, the even the utilization of AI can find in drug based. I told you, even for drug based, disease based, if you have multiple diseases, and even if one disease, multiple targets are involved. So in all this involvement, okay, can find AI is played a major role in developing the softwares, on developing softwares for as well as developing databases. So if you see this AI-based drug discovery for COVID-19, okay, this is taken from a journal publication. Okay, so like a pipeline of computer-aided drug design. Okay, for SARS-CoV-2 is to AI, that is using a machine learning techniques for classification, regression. So all these things are published here. Okay, we can develop any kind of pipelines with AI for any kind of diseases and disease targets. Okay, so all these things are examples of how we can use artificial intelligence. Now let me take you to this example of AI tools in drug discovery. So deep chem, okay, this is using the multiple linear regression, the multi model that uses a Python-based AI system to find a suitable candidate in drug discovery. And deep neural, sorry, deep neural net QACR, organic potential net, hit texture, delta vina, neural graph fingerprint, AlphaFold database from AI, and 10th, computer. These are popular AI tools in drug discovery. AlphaFold, this is an artificial intelligence enabled kind of a discovery for 3D structure of proteins. You know, many researchers get benefited just because of this AlphaFold, because you know only 2.3 lakh structures are available. 2.3 lakh 3D structures of protein are available in PDB database. We have only up to this, but we have millions of molecules. We have more than 40 to 50 millions of protein sequences. 40, that is 400 lakh structure, 400 lakh sequences we have, but we have just 2.3 lakh structures alone. If so, then obviously we have to rely on the homology modeling, ab initio modeling, or threading, isn't it? When compared to that, this database, which is developed through AI, can predict even the 3D structures of proteins. So one can able to use this database uh, instead of going with the computational method of prediction. Okay, if you don't have any structures, then we can go with the computational method of prediction. And this is another slide mentioning about list of AI softwares. plenty of softwares are provided, and even the hyperlinks are provided, including the type of machine learning techniques. If you want to click deep synergy, so we can get the deep synergy is nothing but predicting anti-cancer drug synergy with deep learning. So like this, we can see for each and every website, we can see the code, we can see the data set, we can see the examples, okay, we can see the paper, okay. So we can see everything for each and every kind of a software, okay. So for this, we one can able to go to this software link, and obviously, we can. I'm going to paste all these hyperlinks in the WhatsApp group. Just follow that also. Now let me take you to this list of AI tools. 1 minute. This is list of AI tools. The list of AI tools from Professor Brumiha. I told he is my mentor for my postdoctoral fellow. So this is the website link for him. Okay, if you want to download, I mean, if you want to utilize the tools developed using the AI and machine learning, one can able to go through that. Okay, so anyhow, after the presentation, I take you to these kind of website links. So MPA product, that is a machine learning approach for predicting the binding affinity of membrane protein complexes, and similarly codon de-optimization. So for each and every kind of rare things, whatever we are not practicing, we are whatever is not existing in biological databases, okay, that we can able to see in this database, okay, in his own personal, I mean the ATM page, we can see around 20 to 25 tools, AI tools are developed by his team. So each and every tool is a product of a research scholar, is it's their own PHA research. And even we have nearly 20 to 25 databases developed from the machine learning technique, okay, machine learning and AI. So generally these kind of machine learning technique databases we can able to access through this URL mentioned below. So Indian patent, I just also published a patent in a biomedical imaging system for cancer screening using artificial intelligence. Indian patent. So some of the claims like how we can take the images and how we can go for cancer screening of cancer, okay, and how we can take a different samples and we can able to screen the cancer, and how the different cells are contributed and what are the different risk factors and what are the various medical history data. So all these things I have mentioned in claims. Role of AI in drug discovery. So AI, obviously, wherever you can find the applications of machine learning in drug design and discovery, the similarly even the AI comes there. Okay, majorly AI can use it for prediction of, if you see in drug screening, prediction of toxicity, prediction of bioactivity, prediction of physicochemical property. I think these are all we have discussed in the previous sessions, isn't it? Whatever we are predicting from the tools and databases, okay, this role you can see the indulging of AI inside, okay. And similarly in drug repurposing, in chemical synthesis, okay, like in retrosynthesis pathways, in reaction yield, and okay, and also reaction mechanisms, okay, biospecific drug molecules, designing multi-target drug molecules, where you can find in pharmacology, okay, like this you can find plenty of applications of AI. And some of the tools of AI in drug discovery, deep chem. So most of the tools in machine learning we can able to see in AI. Okay. So fourth one is organic, fifth one is potential net, sixth is hit texture, seventh is delta vina, neural graph fingerprint, alpha fold, computer. So the hyperlinks are mentioned. And this is a figure illustrating AI in cancer research. Okay, so how the artificial intelligence is like, however, I just published a patent of AI in cancer. Okay, so this is something you have to remember, this kind of information. Okay, so more about this, you can follow this URL. I'll be pasting the links for sure. And AI in cancer early diagnosis. So it's most important that if the cancer is diagnosed early stages, okay, in stage one and stage two, it can be cured, isn't it? Then how the AI can indulge inside and to detect initially, okay, in earlier stages. Nowadays, through AI, the detection of AI in early stages is possible. Okay, earlier it was not like that, but now it's everything is possible. So one can see the practical applications of AI in early diagnosis. If you start with stratified screening, recurrence detection prediction, biomarker analysis, image analysis, workflow triage, symptomatic patient triage. Okay, so these are major kind of areas where you can see the AIs can be used for early diagnosis of the cancer. And this is another picture showing about biomedical imaging. So if you take any samples from the slides, the blood samples, or if you take any images from the microscopes, so obviously we take it to the computer and we try to process, isn't it? Whatever we process, okay, we come up with a kind of a classification, regression, clustering kind of a techniques. Okay, whatever techniques we use, as supervised or unsupervised learning. Later we try to find what type of cell, what color of cell you can find in staining, and what is the shape and size of the cell. So all this information, multi-parameters can be used even for diagnosing the cancer and even the stage of cancer. Okay, so each and every information from the cellular point and even the proteins, a cytokine secreted, even these information can also be used to check whether the patients can be treated or not, or what kind of drugs can be used for the stage of cancer. Okay, so here you can see for each and every aspect whatever mentioned, the AI comes inside. And this is another figure from another picture, I mean another journal paper on diagnosis of lung cancer. Okay, how this lung cancer can be detected. So obviously we can use multiple omics, that is proteomics, genomics, transcriptomics, interactomics. All these things can also be used for early diagnosis. If only if, that's because we have everything, all this omics data is present in biological databases. Nowadays, it is easier for us to screen initially. If the patient can come up with a genome, then obviously one can able to find it easily. And even if it could come up with other homes, even the stage and other information can be found out easily, and there can be given an accurate treatments, precision-based treatments for curing that kind of cancer disease. In this case, lung cancer. So breast cancer, for all the cancers, we can see the applications. Head and neck cancer, the Chennai hospitals are famous for treatment of these type head and neck cancer. And A for drug repurposing, I've told multiple examples. So starting with DNA, RNA, protein to interactome, organelles, cell signaling, tissues, organs, organisms, okay, from sequence level to everything. Okay, so we can able to integrate with multiple hidden layers, and but from a image or even from a sequence, one can able to come up with a kind of a base drug for the stage of cancer for a particular patient. Okay, so multiple layers are used, hidden layers are used in machine learning techniques. Hidden Markov model can be used, or the neural networks can be used. So obviously, one should know that AI is come up with a revolution of for a personalized medicine treatment. Okay, so integrating all the types of genome, transcriptome, proteome, phenome, we can able to treat any type of patient. Okay, so this if you go with a breast cancer, it's not a breast cancer. This is something related to COVID-19. So for COVID-19, maybe day 2 patient, day 4 patient, day 6 patient, day 8 patient. If you want to treat these patients based on the stages, even the dosage of drug and the type of drug can also be varied with respect to this kind of treatment. Okay, so precision based approach can be used for the AI. So this is my college. So I'm working in Department of Biotechnology. Plenty of grants we have received from government funding agencies. A database already developed in my lab, and now I'm going to show my UG and PG projects who worked on machine learning techniques for the development of models for the treatment of cancer as well as the hepatitis E virus. So one such important parameter what the student has worked is a receiving operating characteristic. A receiver operating characteristic curve, that is ROC curve, is a graphical plot that illustrates the performance of a binary classifier model. So like we can get the different datas, okay, for true positive, false positive, and true negative, false negative. So there are types of data we can able to get from the diagnosis, isn't it? So if we get these kind of a types of data, okay, how to determine the precision and accuracy of diagnosis of a disease? If only if we diagnose it perfectly, then we can go with the appropriate treatment. So for this kind of thing, okay, identifying the four different types of data, okay, two positive, two negative, so you can see all this. So for all these types of data, one can able to work with the ROC analysis. She has written a Python coding for toxicity feature prediction, okay, and this coding in Python for toxicity processing and ROC analysis, and this is the final output she has received from the ROC. So she has determined, okay, according to the AUC, the random forest algorithm is the best algorithm for our research. And now, if you see this introduction to artificial intelligence, machine learning, and deep learning, okay, artificial intelligence is a broad study, machine learning is a part of a study, and deep learning is, of course, a part of a study of machine learning, and of course, is a part of a study of AI. Okay, this is how we can understand about understand and distinguish about AI, machine learning, and deep learning. Okay, even the information is mentioned here in deep learning, a technique to perform machine learning inspired by our brain's own network of neurons. That's what deep learning. But whereas in machine learning, a technique by which a computer can learn from data without using a complex set of different rules. This approach is mainly based on training data set, that is training a model from data sets. AI mimicking the intelligence. So automatically it should work like how the natural intelligence from human works. Okay, if you want to attend, how much time we can attend? 7 to 8 o'clock, and after maybe we have 2 or 3 minutes extra time, isn't it? So this is how we have the human intelligence to attend the lecture. So whatever we have the human intelligence of, and if the AI is going to mimic exactly at 8 to 8:05, if the window can close, or even in Google Calendar we can set the time, isn't it? Something like 6:30 to 8:15. And participants, sorry for this on today. Unfortunately, whatever Google Calendar link I have saved, it was deleted. I don't know the reason. Okay, and so we have to personally admit the participants. Okay, in the last day, but I think it didn't happen on the other sessions. Okay, so pardon for this thing. I don't know even how to include the kind of a Google Meet link later, because we should not change the Meet link. So it happened, but actually, like even in Google Meet link, we can fix the timing, and even with the after the timing, even we can find that recording everything will try to have a hindering. So this is also a kind of AI. Okay, so even Google has its own AI for finding the uh, patterns, images, or finding the text. For finding the text, we are using Gemini AI, isn't it? For the images, we are using Google Lens. So Google has plenty of AI for recognition of the text, or even the language. So Google also have the AI for detecting the language. Okay, so if you speak in Tamil, it can convert to Hindi. So all this information, so okay, you can able to see this. This is automated. Okay, whatever we think. Okay, if and if you know Tamil and Hindi, then you can able to interpret. But if you don't know, this can automatically interpreting. So this is an artificially enabled human intelligence. So you can see AI is the overall uh, category, and you can find the machine learning and deep learning or part of these subjects. So deep learning, in deep learning we can see ANN, convolution neural network, recurrent neural network, and these are some of the types of data we can use with the deep learning techniques. Types of machine learning I already mentioned the previous slides. And this is another project from my PhD scholar. She has used again a pandas Python programming for hepatitis E virus detection in the patients. So she has written the code and she has taken lot of factors. Okay, and then after taking the lot of factors, she come up with a kind of a precision based information. Okay, so for working on this kind of a Python programming with the machine learning, okay, uh, we have online kind of a tutorials, and we also have a sample code to download that. Okay, so I've already listed some AI tools and databases, isn't it? I can also paste some hyperlinks. In that hyperlinks, you can see plenty of codes, Python programming codes. One can try this Python programming codes. And this is the final result she got. Okay, she classified the types of data from actual and predicted, like a true negative, false positive, false negative, and true positive. So, like the precision, okay, she has determined the precision. That is, the precision is generally predicted through true positive to the ratio of true positive and false positive. So, for the model, whatever she has developed for the logistic regression, she got 87.096, and for the area of under the curve she got 67.98. These are some of the data, statistical data one can able to get from the machine learning technique. Okay, she used the panda programming for determining that. And this is the upcoming certificate and internship course on bioinformatics. This will be conducted from 18th August to 1st September. So earlier registered participants will be getting 10% of the concession fees even if you register individually. But if you register in the form of a group, so obviously 20% is concession you can get. Okay, and these are some of the information one can learn in bioinformatics. And these are some of the organized events in the past 4 years. So we got even sponsorship from the CACR to conduct genomics, data science, and precision medicine design, and are linked with the famous company BioVia company. Okay, so we conducted a kind of a comparative drug and vaccine course. This is the first course I've conducted. Later, we have conducted numerous courses after that. And we also got a kind of a funding from DBT to conduct popular lectures of biotechnology in anti-cancer drug discovery. Later in 2023, we received funding around 3.5 lakhs to conduct an ATAL FTP from AACT. Okay, so the topic is unraveling molecular mechanisms and artificial intelligence approaches of practicing in cancer. I think even I just mentioned, even I published a patent, Indian patent in AI approaches in cancer. Okay, so these are some of the information on AI approaches and AML approaches of drug design in cancer. So if you want to see the kind of a websites, I can take you to the websites. But participants, I want to mention one most important thing, whatever I mentioned in the WhatsApp group. So today I'll be posting the 25 MCQ link, and I will be posting the mandatory Google form for feedback form. So try to fill it up, okay, just for securing the certificate. The marks won't determine, okay, to provide the certificate. Only the attempting provide determines to provide you the certificate. So I suggest everyone to appear to answer for these two forms. Okay. Now let me take you to this kind of links. I can take not more than 2 minutes to illustrate. So first I can take you to the plenty of tools. Okay, AI tools. So this is the link actually. Bio. So if you go to the bioinf.check.at, we have plenty of softwares. Okay, so plenty of software. So the demo tools are there. Okay, so if you go to the particular deep synergy, we can get even the git set data head sets. We have the programming, we have data set, we have programming, we have even compounds examples. We can download it and we can able to work it on. So more about this even we can get from the journal paper reference. Like this, we can try with each and every example mentioned in this website. Okay, deep energy, synergy, podcast, kebabs. So many examples are provided. Now let me take you to the next link. The next link is from Professor Michael Cromia from IIT Madras. So this is the link actually. Here you can see plenty of tools. Okay. So first is for membrane protein complexes. So predicting potential residues for the lung cancer using deep neural network for that can prosite. Okay, different names of that for the tools. PCA PRED for prediction of protein carbohydrate complexes. Actually, I didn't find any kind of these kind of protein carbohydrate complex binding affinity databases, okay, or tools, okay, but we can able to see everything in his in the IIT website, okay. So can see these kind of tools are developed by him, and in resources we can also find the databases. These are the databases developed under machine learning. And myself, I'm an alumni, so I'm a postdoctoral member. So recently completed my PDF in IIT Madras. Okay, now let me take you to the next link. The next link is on the Alpha Fold. I think you know about Alpha Fold. It's there in RCSP PDB, but we also have an exclusive link for the Alpha Fold. Okay, so if you type alphafold.ebi.ac.uk, so if you want to type for a particular protein, give the keyword of a protein, and you can search yeah, the 3D structure. Okay, so you can able to download the 3D structure of the proteins here. Now let me take you to the blogs. Okay, if you want to check with the blogs, okay, of how the TensorFlow is linked with the MATLAB. Okay, so even we can get a stepwise guidance like how to link and how to integrate. So many hyperlinks are mentioned here. So like using the model, converting model, running TensorFlow. So we have hyperlinks and we have journal papers where you can see much of AI and machine learning approaches in this. Okay, so all these hyperlinks I'll be sending it to you today. Any doubts you can ask me. I have completed the last day session because too voluminous. Even if I want to take a particular machine learning algorithm, okay, for this itself I need at least 2 or 3 days, 2 or 3 sessions. So ANML is a ocean. So before going to ANML, one should know what is drug design, what is vaccine design. If only we know this drug and vaccine design, we can use the kind of programming code like Python programming, R programming, and the MATLAB programming, okay, to create the models and to come up with a kind of intelligence in the form of artificial intelligence. Okay, so this is stepwise development to bring any tools or databases for our society. Okay, so I think I have completed the last day session. So if you have any doubts, you can ask. So this is the last day. Anyone can ask and anyone can interact. I can spend up to clearing the
Good
doubts.
good evening, sir.
Good evening, madam. Tell me.
This is Archana Sawant. I would like to ask you a personal research question. Is it okay?
Yes, please. Anything you can ask related to our course.
Yes, sir. Could you direct me to publicly available data sets which contains clinical and outcome data from breast cancer drug trials? I'm particularly interested in data set that will include patients' demographic, treatment protocol, biomarker information, and clinical outcomes.
Madam, actually, myself, I have learned this kind of information from Coursera. In Coursera, we have around 6 courses, and we have even for precision-based kind of course, precision-based medicine. Okay, so where I have download these like whatever you ask, the clinical data sets, and the stepwise guidance is provided in the Coursera itself. If you want a kind of a certificate, we have to pay for the Coursera. But if you don't want, if you want to learn up to the final session, like downloading the data sets and practicing with
Yes.
any of coding, it we have in Coursera.
Okay. Okay.
Yeah, it's very easier for us. Okay, stepwise guidance is provided. We have nearly 2 week session, 3 week session like that. So we
Mm.
have to learn. Yeah.
Actually, sir, I'm working with a prediction model for adverse drug reactions in breast cancer patients.
Oh, good, great. But first we have to work with the sample data sets, like how you
Right,
asked.
right, sir.
Then we can whatever we create, we can able to work.
Yes, sir. Yes, sir.
And any other doubts from any other participants?
Thank you very much, sir.
Thank you, madam. Thank you, Archana. Yeah, any participants can also enter. Not only up to the ANML. If you have any other doubts in the previous 13 sessions, you can ask. So if no doubts, you can integrate with me. I mean, you can keep in touch with me. I think I can also paste once again my LinkedIn profile. Okay, you can keep in touch, and whenever you want any kind of consultancy service, if you want to work on computational part, if you want to work on in vitro cancer cell lines, if you want to work on in vivo animal models. Okay, we have facilities in our institution. We can integrate in the form of consultants. If you want to do PhD research, you can get also stipend. The scholars can get stipend, and even if you want to complete a postdoctoral fellowship, so we can write proposals for AICTE. So AICTE is guiding, also providing a decent kind of a stipend, okay, around 80K. So one can able to register any PhD and PDF under my guidance in any of these AI and ML approaches, as well as in any kind of wet lab studies. Okay, thank you very much for to all the participants who given me the continuous motivation and support to guide these kind of a courses from the TPIT and MSME recognized AST and MIC sponsored startup. So thank you very much, and so kindly try to uh, answer for the feedback as well as for the MCQ questions to secure the certificate. Any doubts, you can keep in touch with the WhatsApp number. Okay.
Thank you, sir.
Thank you. Good night to everyone. I share you the video.